started. And this is our third lecture together. So just, again, a little bit of background. If you want to get a hold of me, I'll be holding office hours on Monday from 1 to 3 online. The Zoom link you can find in Brightspace. If you can't make it at that time, just send me an email, and I'll either try to answer your question by email, or we'll set up an appointment at another time. Okay, so in the last class, we talked a little bit about microscopy, and we're going to continue with that today. And we looked at the two early figures that we associate with microscopy, which are Robert Hooke, who had what we would call a modern microscope, so a compound microscope, meaning it has multiple lenses, and Antony van Leeuwenhoek, who had a very simple microscope that had a single lens, although he actually attained better resolution with his microscope due to the better quality of his optics. We talked about what we would call the cell doctrine, and that's that all life is made from one or more cells, and that the cell is the basic unit of structure for all organisms. And thirdly, we added in this that all cells arise from pre-existing cells. And then that led us into a conversation a little bit about things like artificial cells that are coming out of the J. Craig Venter Institute. We did a review of sort of your first year biology, looking at the 10 important universal features of cells. And finally, we talked about really where or what gave rise to cell biology. And we looked at these three fields of genetics, cytology, and biochemistry, and how they combine to make the field of cell biology. Okay, so, oops. All right, so today we're going to look at light microscopy really briefly. We'll talk about, and then we'll talk in quite a bit of detail about what's called fluorescence microscopy. Okay? And we'll include in that a talk about reporter genes, which is probably one of the more complex things, in fact, that we'll be talking about during this term. It ties together a lot of different concepts. So if we're kind of looking at what we're capable of seeing, we have this chart here, and on this chart, maybe I can outline it here. So if we look at this chart, the top section here, everything kind of above this line, which is around 100 to 200 micrometers, this is what we sort of the average human eye can see. And we can see here that we can see some single cells. So here we have some frog eggs and like a chicken egg, and we can visualize those. But most of the cells we're interested in lie down here, which is below our limit of resolution. So we can't actually see these things. So this is an animal cell, which averages for a human about 50 micrometers. And so in order to see these then, we need to utilize some form of microscopy. And if we use light microscopy, we can see down to approximately 200 nanometers or so. So that's 1,000-fold better than the naked eye. Something I need to point out, you should be familiar with the terminology micro, milli, nano, and you should know how to convert between the two. I assume you know how to do that, and those questions do come up on tests. So with a light microscope, we can see many of the features of a cell, including things like most of the organelles, but we can't see big macromolecular machines. We can't visualize the membranes directly because they're too small. So to see these, what we utilize is what's called electron microscopy. And for this, we can see down to about 0.2 or below micrometers, okay? And so now we're getting into a very, very small objects that we can see with an electron microscope. So 1 angstrom, that unit we have here, is equal to 0.1, oops, sorry, that should be nanometers, okay? And so we can start to see down into the range. It's about, we have 200 picometers here, about 0.2. That's utilizing very specialized kinds of microscopy. The idea, and I was stressing this, we talked about this a little in 200, micro 200, we have two things we need to do. The first thing we need to do is make the object bigger so we can see it, and we call this magnification. All right, so that's the first step. But we can make something bigger and bigger and bigger, and you don't get more detail. And you can think of your phone. You take a picture, and maybe you're whale watching or something, and you see, you know, a whale off in the distance. You can blow that up, but you won't necessarily get improved resolution. And so what we need to have, in addition to magnification, is resolution. And that's really what's going to be limiting for our ability to visualize things, the resolution of the microscope. And so for resolution, that is the minimum distance at which two objects can be distinguished. Okay? So it's the minimum distance at which two objects can be distinguished. If we look at the left-hand image here, is this two things or one sort of blobby H-shaped thing? We can't tell. If we look here, this is probably approaching the limit of resolution. We can see a little gap here. So this is our limit of resolution. And here we can see they're clearly resolved. And so the idea for resolution is we want this to be as small a number as possible, because that means two things that are very, very close, we can tell they're two things. Okay? And so when we're looking here, this also defines our limit of resolution. So for a light microscope, the limit of resolution is about 200 nanometers. And we can show this in a cartoon. Here, this is taken from an old Futurama episode where you want to enhance the depth sphere. Why is it still blurry? That's all the resolution we have. And making it bigger doesn't necessarily make it clearer, okay? So again, we can blow something up, but without the resolution, it's not helpful. We can call that empty magnification. We're making something bigger, but we're not actually seeing any improvement in the details. We can define resolution using this equation, 0.61 times lambda, which is the wavelength, okay, the wavelength of the light, divided by the numerical aperture. And this is a property of the lens. And it's to do with this angle, which they call alpha in this textbook, but you usually see it as theta. So in this particular example, this would be n sine alpha, which is how far that objective lens is from the sample. It makes an angle for the light. And then n is the refractive index of what the light is passing through. And so when you're doing microscopy, you have two things that it passes through: air, which has a refractive index of about 1, but you also sometimes do oil immersion microscopy. And you may not have seen this before. You may have seen it in high school. I don't know if you did demonstrations with it in the first year online labs. Did you? Oil immersion? Yeah? I got some yeses, some nos. Great. So oil is around 1.5. What we want for this value of resolution is we want the number to be very small. And so we want the numerator to be small, this up here. And so we use a short wavelength of light. Okay. The shortest being violet, which we have on the next slide. That's the shortest visible wavelength. There's shorter wavelengths, but the human eye cannot pick them up, such as ultraviolet. And then we want the denominator to be big, so we want as big a thing as possible. So we can see on the right, here we have a greater angle of alpha, theta. If we put this in oil, we have a bigger number. And so, again, this is what we're looking for in resolution. When I talk about high resolution, we need to think what a microscope with a high resolving capacity, that resolution limit will actually be a small number. So you got to flip your mind a little bit here. Okay, if something has very good or high resolution, this value will be very small because that tells us two objects that are very close together, we can tell them apart. Okay, so that's just a little thing if you see on an exam, which will have the highest resolution, and I give you some different combinations, you need to be thinking of the smallest number. Are there any questions about that? Yes?
What does theta mean again?
So theta is what they're showing as alpha here. So this is actually the angle at which the light, it's actually the angle at which the light approaches the lens, the widest point of it. And so one of the things, again, you're maybe not familiar with working directly with the microscopes, is as you go to a higher objective lens, it moves, the lens moves closer and closer to the sample until in oil immersion it's right on top. So that has a really wide theta angle because the light's moving like this. Whereas when you use the lower objective lenses, the light's moving at a smaller angle. Okay? And it has effects on working distance and other things like that that you'll learn about in the labs, but that we won't cover here. Okay. Any other questions? Yes?
So which lens has better resolution, on the left or the right?
So the one on the right will be a higher resolution lens. And when you actually are in the labs, it depends on the microscope. Sometimes the NA, the numerical aperture, is actually stamped right onto the lens, the objective lens. So when you're in the lab, you might want to look and see, I can't remember whether the ones in the 225 lab have that, but it'll actually have the numerical aperture stamped on the objective lens. The objective lens, remember, is the one you sort of flip in, the three on the part at the bottom of the microscope tube. Yes?
So when you do this calculation, is it the distance that you get?
That'll give you a distance, yeah. Depending on what you input as your wavelength, but usually we measure wavelengths in nanometers. So this will give you a distance in nanometers. So what does this mean? Let's do an example here. This is actually, I really like this example. So if we were to think of light as a wave, we can imagine two people that if you stand at opposite ends of a skipping rope and you flip it upside down, you can set up a wave. And if that's our wavelength of light, and we want to see something with it, if we have a long wavelength, like we have in the top example, okay, here we have a long wavelength, something big like a beach ball, if you throw it through the skipping rope, it's likely to hit. But if you throw something smaller, like a softball, it might go through without being visualized. If we shorten the wavelength, as we see in the second to bottom panel, shorter wavelength, what happens here is now the baseball is more likely to hit the skipping rope. And so when we're looking at visible light, This is the shortest visible light, is violet, is about 390 nanometers. Whereas when we're looking at electron microscopy, the wavelength is actually 0.004 nanometers, so much shorter. And so this gives us a much higher resolving capacity on an electron microscope. I'm going to just briefly describe three, how many are on here? Four types of light microscopy. And we're going to really focus our efforts onto talking about fluorescence microscopy because we see a lot of that in this class. The kind of microscopy that you deal with in the labs and that you've probably done before is called bright field. And what happens here is that you have a light below your sample and the light shines through your sample. Some of that light gets blocked, absorbed by the sample. And so what that does is that allows you to then visualize all of the areas where you have material that's dense enough to absorb the light. What's the big issue, and we can see it here with this type of microscopy? You can't see much, right? Because most cells are translucent, meaning the light moves more or less through them, and it's not highly blocked. So what we do is we add stains to these, usually. If you're working in a lab with a bright field microscope, you usually use a stain with it, okay? And what a stain will do is that will block the light and allow you to visualize it. And so the nice thing about stains is that we can develop stains that are specific for certain cellular features, okay? We can often use more than one stain at a time on a sample. And depending on the stain, we may be able to do this with living cells. So we don't necessarily have to kill the cell to stain it. Sometimes we do, but some stains we can use on living cells. So this is a type of microscopy that you're reasonably familiar with. The other two that we see quite often in this course are two things called phase contrast and differential interference contrast microscopy. We're not going to spend a lot of time talking about it, but I'll briefly sort of describe how these work. What happens when light moves through a sample is that if we're looking directly at that light, we can see in the top left corner, there's not a lot of, you can't see many features of the image because not a lot of that light is absorbed. We can increase that through staining. What happens as light moves through a sample, however, is other properties of it are changed. And one thing that changes, maybe I should go to a whiteboard for this, is that if we have a sample here, and we have light, and here's our light, and it's moving through, we know that light travels in straight lines, right? And that's as close as Doug can come to drawing a straight line. So it would look like that. That's how light travels. But when it actually hits a sample, it can be refracted and actually bent. Okay? And we call this, it's refracted. And this is why when you have a spoon or something in a glass of water, and you look at it from a certain angle, the spoon looks like it's broken. Okay? And so what we can do is this light that goes straight through, because also some light might not hit the sample. Don't worry about that weird, PowerPoint has like a terminal glitch that you can't fix where it puts straight lines in when you're drawing, which is fantastic. So we're not really interested necessarily in the light that goes straight through, because it might be light that either nothing has happened to it in the sample, or it's missed the sample, okay? People good with that concept so far? So what we can do is we can actually set up microscopes so that we only look at this light. There's tricks we can do because any light that's refracted means it's hit the sample, okay? So we can set up using optics ways that we only look at the refracted light. Okay? And so what this allows us to do then is only look at the light that has gone through the sample and been altered. And the other thing that alters when light moves through is there's two other important properties of light, and one is called phase. And in phase, let me see if I can draw this, because I don't think I put a slide of this in, because it seems to work better just teaching students about it. I'll do a different color so that there's a lot of drama in this class. So if you look at, that's great. If you look at waves, so here's wavelengths of light, and these are in phase. This is how light will travel if it's from a single source, so that the peaks and the valleys line up. Okay? When it moves through the sample, however, what happens is that they move out of phase. Okay? So you get what's called a phase shift. Okay? And what that phase shift means is that the light cancels itself out. So there's a lot more complication to this, but the long and the short of this is that we can take advantage of the shifting phase of light, and we can do what's called phase contrast microscopy. So this has no stain in it, but look at how much detail we can see. So this can be done using living samples, and it doesn't require any stain. You could add stains to it if you, say, wanted to see a specific feature of the cell, but you get a lot more detail. So what this is doing is this is just increasing the contrast. Similar to what you might do with a digital photo, you can increase the contrast to get additional detail. Differential interference contrast microscopy is also called Nomarski. And we see quite a bit of this in the textbook, Nomarski and phase contrast. Nomarski takes advantage of the change in phase, but also what's called polarization. So you can align the light so that it's traveling in a plane, so a sheet, and when it moves through the sample, that plane tilts. And you can take advantage of that as well. And that's the additional feature for differential interference contrast. It uses phase as well as what we call polarization. And this is how polarized sunglasses work, because most of the light that's reflected into your eyes is traveling in the horizontal plane. So you set up a grid that doesn't let that light through, and that gives you the good contrast so you can see through the surface of the water, for example. This one looks three-dimensional. Those look like little fried eggs, but it's not actually a three-dimensional image. It's a total optical trick. So it gives the appearance of 3 dimensions, but it's not a true three-dimensional image. We're going to talk about a type of microscopy that does give a true three-dimensional image with information in all 3 axes, but this is not one. It just ends up looking that way for whatever reason. Are there any questions about that? No? Okay, so let's move on to talk about fluorescent microscopy. So fluorescent microscopy is based on fluorescence, and what a fluorescent molecule will do is it will absorb light at a low wavelength, or a short wavelength, I should say, and it will emit light at a longer wavelength, okay? And on the right-hand side, we can see examples of many different fluorescent stains, and we can see, so for example, cyan fluorescent protein absorbs light at, what's that, around maybe 440 nanometers, but what it does is it emits light at, that looks like maybe 480 or 475. So all fluorescent molecules absorb short, write that on here, absorb, see it does that again, which is my favorite thing, absorb short or shorter, emit longer wavelengths. If we go down this list of fluorescent stains here, we'll see every single one of these does that. Absorbs a shorter wavelength and emits a longer wavelength. We can take advantage of this property in a fluorescent microscope using what's called a dichromatic mirror, also sometimes called a dichroic mirror. What a dichromatic mirror does is it works like a normal mirror for a short wavelength, and there'll be some wavelength cutoff. And anything longer than that wavelength will actually pass through that mirror as if it's a piece of glass. Okay? Magic, I know. So the way that we can utilize this in a fluorescent microscope is that we can generate a wavelength at right angles to the viewing tube. We can select for a specific short wavelength, which is called the excitation wavelength. So the shorter wavelength, we can see this at the bottom of the chart, is called the excitation wavelength. And it's at right angles, so it will bounce that light down onto our sample. Oops. So it'll bounce them down onto the sample. Oops, this is what we want. The sample will, the fluorescent molecule or dye will absorb that excitation wavelength and emit this longer wavelength. And when that longer wavelength hits the mirror, it goes through to the eyepiece. All the other light is excluded from traveling to the eyepiece, okay? Meaning that some of that blue light we first put on, some of that's going to reflect off our sample probably, right? But it's just going to be bounced away and you won't actually see it. And so by combining different excitation wavelengths with different combinations of mirrors and different fluorescent dyes, we can visualize several different fluorescent dyes on a single sample. Again, sometimes this can be done with living organisms, and we can develop dyes that are specific to different, say, different cellular features. Okay? So the key here, what you need to keep straight, is there's a shorter wavelength, the excitation wavelength, and that's bounced by the mirror because the mirror reflects it. It's absorbed by the fluorescent molecule, and a longer wavelength is emitted, and that's the emission wavelength, and that passes through the mirror to your eye. So this is an incredible tool for cell biology, and you'll see this used a lot, and here are some nice examples. So what we're looking at in this particular example, and I just picked this one because it was done on the International Space Station, which is sort of neat that they're doing this up there. In this particular example, they're looking at 3 different fluorescent markers. One stains the nucleus, which is the DNA component. It's actually staining the DNA of the cells. We're looking at a cell culture here. The second and the third one are looking at 2 different components of the cytoskeleton, and this is the protein framework within a cell. And what we can see is we can look at them individually. So the blue, we're looking at the genetic material. Then we look at the green, which is showing what's called vimentin, which is something called an intermediate filament. And finally, in red, we're looking at something called actin. And what we can see in the end is we can overlay all 3 of these and get a really nice picture of what these 3 specific features look like with respect to each other inside the cell. Okay? And so, again, this is a really powerful tool. All right, there's another sort of feature here which is really important for fluorescence. And for this, I'm going to draw your attention to the side here. And if you look at the side, there's a little teeny tiny blue LED, okay? And if you're in the far corner of the room, you can probably see that. You can see that from pretty much anywhere in the room. You could probably see that if you were standing another 100 meters away. Okay, people are all in agreement with that? So far, so good, no fake news, nice. If that light burned out, and you were in the far corner of the room, or 100 meters away, you would no longer be able to, you wouldn't see the little black dot where it was sitting, okay? If you were me, you might not even see the big white part. So what's important here is that is emitting light. And so it allows you to visualize it, okay? Even though it might be below the limit of resolution. So this is the first important thing here. And this is how we can see stars. You go out at night, you look up, you see a star. A star is far too small for you to see with the naked eye, but because it gives off light, you can see it. What this means is that if you're utilizing fluorescence, you can use it to view things in the cell that are below the limit of resolution, that you normally wouldn't be able to see. Okay, so that's the first step. The caveat here is if I'm standing 100 meters away and I see this blue light, and even looking from here, is that one blue light? Or is it 4 little blue lights that are very close together? Is it 6 little blue lights? I can't tell. I can just see the light. So what happens is you can increase the detection, but not the resolution. And if we look at this diagram here, it displays it really well. The arrows are just sort of focusing you on different parts of the cell. But if we look in here, for example, so on the left-hand image, we're looking at a resolved image taken through, this is taken through a form of electron microscopy. And we can clearly see, well, you can probably see it on your notes, maybe not up here. But there, we can see well-resolved, again, this is looking at cytoskeletal elements. And so we can see them individually. If we move down to a section where there's more of them, we can resolve sort of that there's, it looks like, maybe 4 or 5 cytoskeletal elements there. If we look on the right, we can detect these. Cytoskeletal elements they're too narrow to actually visualize by light microscopy. But if you utilize this fluorescence, we can see them, but we can't resolve them. So, how many are in this bundle here? We can resolve them on the left, but we can't resolve them on the right, although we can detect them, okay? So that's another really cool feature of fluorescence microscopy. It allows us to see very, very small things. Okay? Any questions about that? Okay. Let's move on. It gets more and more exciting as we move through it. This is something called green fluorescence protein. And green fluorescence protein is a protein that's produced by a jellyfish. It's a fluorescent protein, meaning that this protein itself will absorb an excitation, shorter wavelength of light, and emit a longer emission wave of light. So it absorbs blue light and emits green light in this example. If we think back to our 10 universal features of cells, one of the cool things we remember is that we can take genes from one organism and put them in another organism. And so there is a gene in the jellyfish that encodes for this protein called green fluorescent protein. And the other thing we know about proteins is that they're actually produced by the cell. So with other fluorescent stains, we have to, if it's something we want to stain inside the cell, we have to find a way to get that stain inside the cell. Here, what's happening is the cells can actually produce the protein naturally. And so the cell itself produces the fluorescent molecule that we can use for different things. And this is going to be the trickiest thing we look at in the first part of the course. We can use this to assemble what are called reporter genes. And we're going to focus on the top left panel here. We're not going to talk about fusion proteins in this class. If it's something you're interested in, I'm happy to walk you through it in office hours or at another time. It just adds a sort of an unnecessary level of complication that we don't need at this point. But reporter genes, I think, are important for reasons that we'll get into. Okay, so I do have, if you go through last year's lectures, I do have a pretty detailed lecture of this that you might find helpful. But I will explain this to you now, and we'll do the best we can. So at this point, you're at a little bit of a disadvantage because your knowledge of genetics, you haven't done 230 yet, so you might need some reminding of it. And so I'm going to give you a really brief overview then of genetics. And so this is DNA. Pretend this is double-stranded. And what you have here is a gene. We're going to draw that as a box. And we have, remember, our transcriptional start site, which we call +1. And remember, this region that's the box, through the process of transcription, will become an mRNA that corresponds to the length of that box of the DNA. And then through translation, will become a protein. Okay? So we have a lot of different types of cells. And one of the major things that determines the function of each cell are what genes are being expressed. Okay? And so we're going to do this as simply as possible. But what we have is we have a number of factors, transcription factors, activators, repressors, and what these do is they determine whether this gene, the red gene, is on or off. And they also determine how much it's on and off. Okay? So let's say I am really interested in red gene. We'll just call this red gene. And I want to know within my cell, I'm growing, or I'm looking, let's say I'm actually looking at tissue from a mouse. And I want to know where red gene is expressed. That might be very complicated to do. Okay? Red gene might not be easy. It might not be easy to figure out how much red gene is in each individual cell. Okay? And so it's hard to measure. Even if I know how to measure it, it might be something that takes a lot of time or is expensive to measure or needs specific reagents. And so there's lots of reasons it might be difficult for me to directly measure how much red gene is being produced. Okay? People are okay with that? All right. So what determines how much red gene is produced? Well, the DNA itself has sequences that will bind to those different factors, okay? And so the DNA itself has sequences which will bind to factors that are either going to turn red gene on or red gene off. So let's say this green, let's not use green because we're going to need that in a minute. Let's say this purple, if the purple block is present, that will bind to the DNA and that will turn on transcription. Okay? And so this is a drastic oversimplification, but often the regions where these factors bind are just what we call upstream, so just before the start of the transcriptional start site. Okay? So this region here. And if this binds a purple block, it's turned on. Okay, but if it binds to a purple circle, it's turned off. Okay, so these are on-off signals, for example. Now, so I want to know how much of this is being produced, right? So what I can do, but I can't trace red genes. So what I can do is I can do a little trick here, and I can make what's called a reporter gene. And what I do is this region here, which is where all the regulatory stuff is, I construct a gene that has that. So let's just mark this with sort of little hash lines so you know. So this region here, from red gene, I cut that out. And this is my +1 site. So this region here will bind to the purple square, and that'll be turned off. Or the purple square will be turned on, and the purple circle will turn it off. What's cool about a cell, if we think back, is the cell actually doesn't care what's after the +1. So what I do is, instead of red gene after it, I put the gene for my green fluorescent protein. So now, if my cell has purple square in it, what am I going to see? I'm going to see that the cell fluoresces because it will be turned on. And so we call this a reporter gene because the amount of GFP that's produced is equivalent to the amount of, in this case, red gene that's produced, because they have the same regulation. The regulation is the same. The way we make this gene is that the cell will still have red gene in it because red gene might be required for that cell to survive or to do whatever its function is. And then we add the bottom one, the reporter gene, in additionally. So they're actually both in there at the same time. And the idea then is that however much red gene is being made, if there's lots of purple square in a cell, lots of red gene will be getting made, and also lots of GFP, so it's going to glow. If there's no purple, or there's lots of red circle, red gene won't be getting produced, and neither will GFP, so those cells will be dark. And if we put that together, we can see on the left a great example of this. So here we have a reporter gene that is specific to neurons, so cells of the nervous system. And only cells, neuronal cells, that have that combination of on factors will produce this, what's normally a neuronal protein, but they also now are producing GFP, and we can see this is in a Drosophila embryo. We can see the entire developing neural network because it glows green, and all of the surrounding cells, all of the tissues surrounding it are dark because they don't have that right combination of factors to turn the gene on. And so reporter genes then are a really important feature that we talk about. We'll see examples of it later, and you'll definitely see it in your third-year courses. Is there any quick questions about that? Yes?
Did we make that reporter gene, or is it something that we just sequence and just, this is right before
Yeah, so we engineer and construct the reporter gene. We physically do that and then introduce it to the cells. Yeah. Okay, so to give you a few minutes to clear out before the crowds come in, I think we'll stop there. We'll see everybody tomorrow.